A father, standing waist-deep in a public swimming pond in Star, Idaho, watched something drift past his leg last July that he took for a feather. Then it moved on its own. Minutes later, his daughters had 15 or 16 of them in a plastic bucket, and every single one was a jellyfish in fresh water hundreds of miles from the sea. But those jellyfish did not arrive in that pond last July. They had been living in it the whole time, too small for anybody to see, and something switched them on. The pond is the East Pond at Freedom Park in Star, a small town northwest of Boise. Mark Brandis was swimming there with his kids when he spotted the first one. He told the Idaho Statesman he thought it was a feather, or some gross mucousy thing floating around, until he realized it was moving. His daughter grabbed a bucket. By the time the three girls were done scooping, they had a bucket full of translucent, coin-sized bells pulsing against the plastic. He did the sensible thing. He called Idaho Fish and Game, and then the Idaho Department of Environmental Quality, and asked whether he should be worried. What he got back was reassuring and, in a strange way, unsatisfying. The jellyfish are harmless. They have been in Idaho a long time. Beyond that, the state did not have much to tell him. That is not the agencies being lazy. That is the honest state of what anyone knows about this animal. The species is called Craspedacusta sauerbii, and biologists sometimes call it the peach blossom jellyfish. According to the National Park Service, a fully grown one measures between 5 and 25 millimeters across, which puts it somewhere between a dime and a quarter, and it can carry up to 500 tentacles around the rim of its bell. It is the only jellyfish in North America that lives in pure fresh water. And it is not confined to one strange pond in Idaho. The United States Geological Survey keeps the national database of non-native aquatic species. Their profile for this animal lists it in 44 states and the District of Columbia. 44 out of 50. Ponds in city parks, gravel pits, quarries, farm reservoirs, slow backwaters of the Ohio River and the Tennessee River, drinking water reservoirs, and ordinary lakes with houses and docks around the edge. So, the obvious question is how an animal from a river valley in China ended up in a swimming pond in southern Idaho. And the answer to that starts in a greenhouse in London, in the summer of 1880. The Royal Botanic Society kept gardens in Regent's Park, and in one of the glass houses there was a heated tank growing Victoria regia, the giant Amazon water lily with pads wide enough to hold a child. Staff noticed small transparent bells pulsing in the water around the lily pads. The zoologist E. Ray Lancaster examined them and published a description in the journal Nature in June of that year. The World Hydrozoa database still lists the type locality of this species, the official place where science first met it, as a tank in the water lily glasshouse at Regent's Park, London. So the first freshwater jellyfish known to Western science turned up in a heated planter, in a greenhouse, in the middle of a city, thousands of miles from anywhere it belonged. That detail is the whole explanation. The jellyfish came in with the plants. Imported aquatic plants for water gardens, ornamental ponds and aquariums carried something almost nobody could see, and the plant trade shipped it around the world. That is the vector the Smithsonian Environmental Research Center points to for the London record, and it is the same route the National Park Service and the USGS point to for how the species reached North America. Wisconsin's own accounts say the same thing. Plants came in for water gardens and aquariums, then reached open water through flooding, or through somebody emptying a tank into a pond. Which raises the question that the rest of this video is really about. If the jellyfish arrived on plants, and the plants arrived over a century ago, why does a lake that has never had jellyfish suddenly fill with them for 3 weeks in August, and then go back to having none for the next 20 years? The answer is that the jellyfish you can see is only a few weeks of this animal's life. Almost all of it is spent as something else entirely. That something else is a polyp. It attaches to submerged plants, to rocks, to sunken branches and tree stumps, and it does not look remotely like a jellyfish. Research published on the species in Italy puts the polyp at roughly 0.5 millimeter to 2 millimeters tall. Other work describes them as, at most, about 1 millimeter. They have no tentacles. They sit alone or in small clusters of 2 or 3, and they are effectively invisible in a lake. You could take a handful of pondweed out of the water, hold it up, look straight at 40 of them, and see nothing but plant. The polyp does not need a mate. It reproduces by budding. It can bud more polyps. It can bud small, crawling propagules called frustules that creep a short distance across the bottom and set up as new polyps. And, under the right conditions, it can bud jellyfish. When winter comes, the polyps contract into hard little resting bodies, and they wait. That is the animal. A speck under a fingernail's thickness, sitting on a rock, capable of waiting out years. If that idea bothers you, subscribe, because that is the entire subject of this channel. What is actually under the water you swim in, and how little of it you can see? And that waiting strategy is not some accident of being transplanted. It is what the animal's home river built it for. The USGS description of its native range is worth hearing. In the Yangtze Valley, this species lives in shallow pools along the river, and those pools live at the mercy of the main channel. Water levels rise and drop. Temperatures swing. The plankton it feeds on appears and disappears with the season. Something living in a pool that might be full in June and half gone by September has two options. It can be fragile and get wiped out, or it can spend almost its entire existence as a speck that is very hard to kill and gamble everything on the good weeks when they arrive. This one took the second option. Then the plant trade picked it up and dropped it into a country full of farm ponds, quarries, and reservoirs that behave, from a polyp's point of view, a great deal like the Yangtze floodplain. And here is my favorite proof of how completely invisible that stage is, because it did not just fool swimmers. It fooled the scientists for almost half a century. In the 1880s, a naturalist named Edward Potts was working in Tacony Creek, a tributary of the Delaware River just outside Philadelphia. He found a tiny tentacle-less hydroid on the creek bed, and in 1885 he published it as a new species, in its own genus. He called it Microhydra ryderi. Meanwhile, the jellyfish from the London greenhouse was catalogued as Craspedicusta sowerbii, in a different genus entirely. Two names, two supposed animals, two separate entries in the scientific record. They were the same creature. Nobody proved it until 1928, when a paper in the Proceedings of the Zoological Society of London finally established that Potts's tiny Philadelphia polyp and the Regent's Park medusa were two stages of one species. For more than 40 years, professional zoologists holding both forms in their hands with microscopes filed them separately, because the invisible stage looks nothing like the visible one. If the experts could not connect those two things for 4 decades, the odds that anyone noticed them in their local swimming hole are basically zero. So we have millions of invisible polyps sitting in American lakes. What flips the switch? Warm, calm water, mostly. The medusae show up in late summer and early autumn. Reporting on the Minnesota sightings notes that in warmer parts of the world, blooms are linked to summer water temperatures around 84 to 86 degrees Fahrenheit. A recent study of Michigan lakes found abundance peaking in mid-September, with a thermal optimum closer to 77 degrees. Indiana's Department of Natural Resources tells people to look in August and September, in calm water, in the afternoon or early evening on clear, warm days, when the medusae rise toward the surface. When conditions line up, the polyps start budding jellyfish instead of more polyps. Those jellyfish grow, swim up into open water, and a lake that had nothing in it 3 weeks ago now has hundreds of pulsing bells in the top few feet. Now here is where the honest version of this story separates from the tidy version. Temperature tells you roughly what time of year to look, and that is about as far as it goes. 2 lakes in the same county, same heat wave, same August, and one of them blooms while the other one does nothing. The same lake blooms one summer and then stays empty for the next 11. A study of detection methods for this species states it plainly. Medusa blooms usually will not occur annually in a given system, and the animal may sit there as undetected polyps for years before any jellyfish are seen. And it is not that nobody has looked. A research paper published last year, working on culturing the full life cycle in a laboratory, states outright that the conditions required for growth and reproduction of the different life stages and the environmental cues that regulate the transitions between them are still not fully understood. A survey of Michigan lakes described the unpredictable nature of these blooms as the central difficulty in tracking the species at all. Indiana's DNR does not hedge. Their appearance, the state says, is sporadic and unpredictable. So the season is about the only part of this anyone can predict, and even then, only loosely, because nobody can tell you the year and nobody can tell you the lake. There is one more piece of biology here, and for my money, it is the strangest fact in this entire story. Go out to a lake in the middle of a bloom, net 100 of these jellyfish, and check them. In most American populations, they will all be the same sex, all male or all female. Animal Diversity Web, run by the University of Michigan's Museum of Zoology, states that most populations are strictly one sex. The pattern is well enough established in the research literature that a 2022 study on temperature and this species cites earlier work, noting that in invaded habitats, the life cycle often is not completed at all, because male and female medusas so rarely turn up together. Think about what that means. A polyp buds by cloning itself. Every jellyfish that comes off a colony carries the same genetics as the colony that made it. So a bloom is usually one animal, copied a few hundred times, all released in the same couple of weeks. A whole lake of jellyfish, and in a lot of cases, not one of them can breed with another, because genetically they are the same individual over and over. That is the mechanism. Now watch what it looks like when it happens to a place people actually use. August 2022, Louisville, Kentucky. Freshwater jellyfish turn up in the Lily Pond at Shawnee Park on the west side of the city, a public park with a walking path around the water. The Louisville Olmsted Parks Conservancy posts photographs. It is the first time the jellyfish have ever been reported in that pond. Park staff suspect waterfowl carried them in, hitching a ride on ducks between bodies of water, and note the jellyfish are almost certainly in the Ohio River nearby too, just far harder to see. Then winter. Then the obvious question of whether they would ever come back. June 2023. Hundreds of them. Back in the same pond, described by local reporting as roughly the size of a contact lens, pulsing up and down in the water where people walk their dogs past every evening. Now, Wisconsin. In October 2014, a freshwater jellyfish was confirmed in Spencer Lake, in Waupaca County, south of the city of Waupaca. What makes that one worth naming is the count that came with it. The Wisconsin Department of Natural Resources logged it as the 94th body of water in the state known to contain freshwater jellyfish. 94 in one state. And the DNR's records of them in Wisconsin go back to 1969, more than half a century of quiet presence in lakes. People fish, water ski, and swim in every summer. That particular jellyfish was spotted in 30 to 40 feet of water. So if you have a lake, a pond, a quarry, or a stretch of river you have swum in your whole life, put it in the comments. Name the state and name the water. This species has been reported in enough places that somebody reading those comments is going to recognize their own. Then Minnesota, and this is the one that shows you the scale of the blind spot. Late October 2025, Taft Lake in Richfield, a 12-acre lake in the Minneapolis suburbs, sitting practically under the flight path of the international airport. Two water scientists from an engineering firm were canoeing across it, doing routine water quality sampling for the city. Kate Lucas saw a flicker of light rise past the canoe and yelled. They netted 2 freshwater jellyfish and preserved them in ethanol. The state's invertebrate biologist told local reporters there are maybe about a dozen records from Minnesota total, and that most years pass without a single verified sighting in the entire state. A dozen records in the whole history of the state. Now hold those two numbers next to each other. 94 water bodies in Wisconsin, about 12 records in Minnesota. Those states share a border, share a climate, share the same glacial lakes. There is no biological reason for that gap. The gap is not jellyfish. The gap is people looking. And we can actually measure that because someone checked. Between 2019 and 2022, collaborators with the University of Maryland Center for Environmental Science ran environmental DNA sampling across National Park Service sites in the National Capital Region, the parks around Washington, D.C. Instead of looking for jellyfish, they filtered water and looked for the animal's genetic traces. According to the Park Service, that sampling produced preliminary evidence of freshwater jellyfish in every single park in the region. Every park. Rock Creek Park, the C&O Canal, Harper's Ferry, Manassas, Prince William Forest. Water that millions of people walk beside, wade in and paddle on. Nobody saw a bloom. The jellyfish were simply there, in the water, in a form too small to register. And the only reason anyone knows is that a lab found their DNA. Which brings us to the question everybody asks about this animal. And I'm going to give it to you straight, because this channel does not sell you a threat that is not there. They cannot hurt you. It is not a matter of them being shy or small enough to avoid. The tentacles are genuinely armed. Each one carries stinging cells, real nematocysts, the same weapon a sea jelly uses. They fire, they work, and they kill zooplankton constantly. The reason nothing happens to you is mechanical. The National Park Service states these jellyfish pose no danger to humans and are simply too small to sting anything as large as we are. Minnesota's DNR puts it the same way. The stingers exist, and they cannot be felt by people or by animals. Wisconsin specialist said it plainly when the Spencer Lake one was confirmed. The stinging cells are harmless to humans. You could swim through 100 of them and never know. That does not mean nothing is happening in the lake. These are predators, and what they eat is zooplankton, which is also what young fish eat. Wisconsin's conservation staff flagged that overlap directly when the Spencer Lake jellyfish was confirmed, noting the species feeds on the same food source as young fish, and may therefore have some impact on fish populations. Minnesota's invertebrate biologist made a similar point to reporters, saying the medusae may briefly reduce the food available to zooplankton eating fish like perch, while adding there is no evidence they disrupt lake use or cause broader harm. That is the real research question hiding inside this story, and it is genuinely open. It has nothing to do with swimmers. It is what a few hundred efficient little predators do to a food web across 3 weeks in a lake that nobody happened to be measuring at the time. And that is exactly what makes this story sit strangely, once you understand the mechanism instead of the headline. Because the animal is not the unsettling part. A quarter-sized jelly that cannot break your skin is not a threat. What should get your attention is what this case proves about the water itself. A lake near your house can hold a living animal for 30, 40, 50 years. It can hold it through droughts, through freezes, through every summer you swam in it as a kid, and every summer you took your own kids there. State agencies can survey that lake. Anglers can fish it every weekend. And the animal sits there the entire time, budding, waiting, invisible, until some combination of warmth and food and something we still cannot name flips the switch, and for 2 or 3 weeks, the lake shows you what was always in it. Idaho is the cleanest example of that I have found. Back in 1997, the Idaho Statesman ran a report on freshwater jellyfish being found in the state by a group of Girl Scouts. That report is still listed today in the USGS reference file for the species. Nearly 30 years later, in the same state, a dad with a bucket and 3 excited daughters found them again, called 2 state agencies and was told, essentially, yes, they are here. They have been here a long time. And no, we cannot tell you much more than that. Nothing changed in that water between 1997 and 2026. The jellyfish were never gone. We were just not looking, and they were not switched on. So, the next time you are standing in a lake in late August, in that flat, warm water near the surface where the light goes green, understand what you are actually looking at. The water is not empty just because it looks it. Everything visible in that lake is a fraction of what lives in it. And the freshwater jellyfish is simply the one that occasionally shows itself for a few weeks before switching back off and disappearing for a decade. The polyps do not leave when the jellyfish do. They are down there right now, on the rocks under the dock, on the weeds by the boat ramp, roughly a millimeter tall, doing nothing at all and waiting for a summer that suits them. And every lake that has never reported a single jellyfish is not a lake without them. It is a lake nobody has checked,